Okay, I'd like to start the second half of our morning program. As you can see, the theme of holistic health is going to follow a few different courses. We started off with mental and behavioral health and well-being as it fits in with cultural health and our ties with the animals, plants, and environment which we live with. And then Dr. DeBass talked a bit about ticks and emerging diseases there. Our next talk is another emerging and very important area of One Health, and that is the concern that we often have with antimicrobial resistant bacteria and how they are present in our environment and may pose a risk to us. And in this particular instance, the discussion revolves around urban moose. So please welcome Grace Leu Burke and join me in thanking her for presenting to us today.
Yeah, they told me the PowerPoint thing.
Yeah, it should be up.
I'm always the shortest speaker. So yes, I'm Grace Leu Burke. I'm from the University of Alaska, Anchorage, and I teach clinical microbiology in the medical laboratory science program, which most of you may not be aware of. I always say you don't know who we are, you just see our laboratory bill. So the one thing that I did when I came to Alaska to teach at UAA was to understand the fact of the One Health model. Coming from Kansas, the concept of One Health is actually very much part of our environment, the animals, agriculture. We are not just a silo. We are what we eat. We are who we are around. And I wanted to bring this model and build it into the UAA MedLab Science program. And why that is because I remind my students that it doesn't do any good to study clinical microbiology and to be in that clinical microbiology lab if you do not know what's coming in your back door. If you are not aware of the environmental and the antimicrobial resistant microorganisms, or as we all now know through SARS-CoV-2, the virus that's linking and sneaking around, you are not doing your patients a service. So the concept of the fact that we are all one, us, the environment, our animals, is really important, and I embedded that into our program. So again, the whole thing with us, I was looking at domesticated animals because, you know, if we're in Alaska, we love our dogs, and we love our outdoors, we love our wildlife, we love our ability to be outside even when it's minus 23. I know I brought my cross-country skis. I mean, this is what we do, but how that works with our healthcare and us in humans. There is a very much potential transfer, and of course, again, we like it, we like being outside. This is why it's important to Alaskans, because we're a big recreation state. So there's always a high risk of combination between us and animals. And the fact that Anchorage lives with approximately 1,500 moose, the fact that I can't necessarily get to my office because the moose is standing right in the middle of my path is not something that's unusual. But we also have a lot of tourism. And, you know, people are coming in, and I tell my students that's like the boat people. You know, they come off those cruise ships and they come into our environment. And what does that mean, and what does that do to our environment? And finally, like any other urban facility, Anchorage is not immune to our housing insecure and our homeless population. And where they live and where they conduct their living is all part of how we are with our environment. Because this vulnerable population is very much something that we sometimes see as underserved. And as we look for our diversity, equity, and inclusion, my students are already recognizing the fact that if we aren't studying the environment where our housing insecure are, then we're not studying the environment of their health and then what they have when they come into our hospitals. So this is why we do it. This is pictures that I take walking around, and I see my bear scat. Yes, there's bear on campus. And moose scat, and there's also garbage, and there's people on the trails, and there's hiking. And this is Anchorage. This is our environment, and this is what I was concerned about, especially that nice pile of moose scat on the sidewalk, and that's actually right near one of our hospitals. And I kept thinking to myself, if this was Kansas and this was my cow scat, I'd all be cited by the health department because we can't do that. But in Anchorage, we just sort of walk around stuff. So what I decided to do is I decided to say, okay, so what is this and what impact does this have on our environment? In this day and age of molecular sequencing is really great, but as a microbiologist, I don't necessarily care if that gene sequence is there. I want to know if it's actually growing. I want to know if I have something that once I have it and it gets in a warm, moist environment, what is left? And so that's essentially what we did. The students at the Med Lab Science program and I ground collected basically scat that's on the ground in snow. We kept it in cold conditions, and then using traditional microbiology, we grew stuff. And we also grew gram-negative and gram-positive. So all you non-microbiologists out here, we got two big classifications, because a guy named Graham figured this out. And we have gram-negative organisms, and that's what you think of your E. coli. And you have your gram-positives, and that's what you think of your Staph aureus. So we also wanted a protocol for gram-positive cocci. Very similar, just a little bit different in how we're going to collect them and how we're going to process them. So here's the students at UAA, and they're collecting these cultures. And what I mean by collecting these cultures is that over the period since 2018 moving forward, we have done over 250 scat cultures. This has provided tremendous experience for them to not only look at what the microorganisms are, but they're actually using the same biological techniques they would use in the clinical lab when they become clinical microbiologists, except they're very good at looking at mixed cultures, looking at pathogens, and for us, looking at multi-drug resistance. We also use something that's called breakpoint values to determine whether or not resistance actually occurs. Microscan and Vitek are two industry leaders in this area, and my students actually use industry-level ability to determine whether or not there's antimicrobial resistance. ESBL is a real concern for us when it comes to gram-negative. That's extended spectrum beta-lactamase. That basically means any of your beta-lactam antibiotics, you know, your penicillins, your cephalosporins, are no longer able to fight that infection. It is a very big concern in the medical community, and so we wanted to make sure to see if this is actually in our environment. So this is a microscan panel. Again, I always know I'm sometimes speaking to non-microbiologists. So you see where that nice yellow stuff is growing? That means that antibiotic isn't working. We actually do serial dilutions to determine exactly at what level the antibiotic will actually cause the organism not to grow. Doesn't look very good, does it? That's a Hafnia species, and all those yellow things mean that's all the resistance. And then the nice colorful things on top, that's biochemical testing, and we know exactly then what that organism is as genus and species, and we know what genes have been turned on, antimicrobial resistant genes and all, because we actually see it on our plates. We also do confirm everything, so no matter what we do, we think, okay, this looks really scary, but we're going to confirm it, especially when we're talking about ESBLs. And again, we're using industry level that we do in the clinical lab to confirm these antimicrobial resistant organisms. And then we have a group that actually looks at all these numbers. So the students do all of this as part of their work in our microbiology labs. They're doing all of this work as part of their training and learning to be clinical microbiologists, but we still have a lot of data. So I have a group of students that actually then look at this data, and this is something that we started seeing. One of the first things we saw is that we saw where we had our culture our pets. You know, we're culturing the ground collection of our moose, and we're going to culture our pets. And we had one of my students cultured his pets, cultured some moose scat that's in his backyard, because everybody in Anchorage has moose scat in their backyard, and discovered that there was multi-drug resistant and ESBL producing E. coli and Klebsiella that were in the moose that were identical to his dog. So in other words, the organism, then the moose scat that he was finding in the yard of his backyard was colonized in one of his dogs. So that was kind of scary, so we started to keep looking. And anything that you see is highlighted. They're just like scanning. This is just like, you know, screenshots of some of the Excel sheets we have. We're just seeing so much more resistant in our Enterobacteriaceae than we ever expected to be possible. And then we saw things that were not only multi-drug resistant, which means more than two classifications of antibiotics. So your beta-lactams, your fluoroquinones, and then maybe an aminoglycoside. But also an ESBL producer, which again makes it even that much more potent. As you can see, these are the organisms we have isolated. If they're yellow and red, that means they're multi-drug resistant. They're also producing an ESBL. Essentially what this means is if you're colonized this, and then you give yourself an infection, which we do a lot, there wouldn't be very many antibiotics that we could use to combat it. So we're really concerned because ESBL transmission occurs. It occurs, publications that show has encouraged from animals to humans, in agriculture, in veterinarian, in food safety, food areas. So we're really concerned that this transmission to humans is not something to take lightly. And then the other thing about the gram-positives is something that's called Enterococcus. So when all of this we're coming up with something that's called vancomycin-resistant Enterococcus. Anybody in here in the healthcare field? Okay, so Enterococcus, you're never going to want to go to the hospital now. So Enterococcus fecalis and faecium are organisms that have true resistance to both an antibiotic that is very difficult then to treat. So something that's vancomycin-resistant Enterococcus fecalis or fecaum means that they received it because you were getting a lot of antibiotics. So you're in the ICU, you're getting a lot of antibiotics, and you get this resistant organism. And then if you become infected with it, it's very difficult to treat. We are finding this in our animals, in our moose, in our bear, and our geese. So again, we're looking at all the wildlife that are defecating in Anchorage, and we're looking at the geese. And we're not surprised with geese. We're not surprised with geese because of the migratory patterns and because we have basically eliminated our wastewater in our nice waterways for our animals to land, that they all use wastewater treatment plants when they're using their open water sources. They're traveling to and from the lower 48 to Alaska. So we're not too surprised that a normal fecal bacterium in humans that can be colonized with high resistance is in our geese. What I was surprised is that I would see it in our moose. And what I was really surprised, and this was we actually spoke at the American Society for Microbiology on this, is that not only were we finding vancomycin resistance, we're finding something that's called linezolid. Linezolid is the only antibiotic we can use when this organism is infected with you. So now I have something that is highly drug resistant, and then our backup antibiotic's not working either. So what does this mean? I was really getting a lot of data. I was getting kind of concerned, and I always love it when I have a student that needs something to do. And so I needed to figure out exactly where we have a human-animal interaction, where we were collecting these samples, where these samples were coming from, and what was our risk assessment. So I had a student do a really great chart, a map here, to determine where our high impact of human and urban wildlife. And we were focusing a lot on moose because it just had we just had a broader collection area, I had a better sampling. So these are where we know we had high impact. And when we looked at this and we talked about it, we figured out there's just 3 different areas. Whether or not there is water, so along our creek beds, around our lagoons, a couple of our lake areas in Anchorage, if it's in a residential or like a trail where there's no water. And then we thought about the areas where we have a lot of, we call it the UMed district. So you have your medical clinics, you have your hospital. And those were 3 areas of urban moose and human interaction that were somewhat different. So our creek beds typically is our unsecured housing and our recreation, where our residential areas like your backyard, where you have more of a day-to-day with your backyard, and of course, if you're going to the hospital, the UMed area. So when we did that, we pulled out 110 samples, moose samples, of what we collected between 2018 and 2020, and this is what we found. We found a significant number of vancomycin-resistant Enterococcus, multi-drug resistant gram-negatives, and some that were multiple. But what's really concerning is essentially 71% of all the samples were positive for one or more drug-resistant organism. 71%. So then we thought, well, is it always all just in the hospital? So we looked at the data again, and yes, there was some significance when we were a waterway and a hospital for vancomycin-resistant Enterococcus, which sort of makes sense because I still think it's probably coming from the migratory geese. But the question is that no matter where you go in Anchorage, whether it's just down a nice trail, along a creek bed, or you're headed to the hospital for something, your risk associated with a multi-drug resistant or ESBL gram-negative bacilli is the same. So this is kind of another visual summary of the fact that of the 110 samples, your chances of having an ESBL, E. coli, or Klebsiella is just equal, no matter where you are in Anchorage. So why do we worry? Well, if you're a clinical microbiologist, ESBL and vancomycin resistant, both of those antimicrobial resistant microorganisms, we have always thought of being nosocomial. We have always thought that they came from our long-term ICU patients on antibiotics. We always thought that they came from somebody that was given antibiotics inappropriately, and now we have this resistance, and now we have to be careful because they could spread it to another patient. Nosocomial happened in the hospital. And I'm telling you, you have a 71% chance of being colonized outside the hospital just because the moose. And then the question is, why do our moose have that? So we are continually looking at this as an ongoing surveillance, and this is only a small part of our project that we're doing. So please, if you're curious or want to know what we're doing, just please shoot me an email at UAA, and I'll gladly talk about this, because we've also noticed not only a resistance, but an increased resistance over time. We had Escherichia coli in 2018 that didn't seem to have any antimicrobial resistance, and we have now isolated E. coli that's resistant to all antibiotics, why is that happening? And I want to emphasize that when I would tell my students when we're looking at this is just not 1 or 2 colonies. This is a Petri plate full of bacteria on a sample that was picked up frozen, kept frozen for at least 6 months until we got around to working it up. Finally thawed it, and that's what grew. So in other words, it's the only thing sometimes that's growing. So I always really appreciate coming here right before lunch to let you know that you got to be really careful what's out there, and really careful when your dog is rolling in that scat in the backyard. I thank you very much.